Let's talk about cannabis. You know, it's got to be one of the most debated plants on the entire planet, right? For some people, it's a dangerous substance to be avoided at all costs. For others, it's a life-saving medicine. So how can it possibly be both? Well, the answer isn't really in the politics or the headlines. It's deep inside the incredibly complex wiring of our own brains.
And right there, that's the heart of the paradox. On one hand, you have a cannabis-derived compound that's an FDA-approved medicine, literally stopping severe seizures in children. And on the other hand, you have study after study linking heavy use to really serious risks, like psychosis. So what is going on? What's the secret behind this double life? Well, it turns out the answer isn't in the plant itself, but in a truly remarkable system that's already running inside you right now.
So, to really get to the bottom of this, we need to meet the brain's very own internal cannabis system. It's called the endocannabinoid system, or just ECS for short. And this is the biological stage where all the action happens, the machinery that the chemicals from the cannabis plant actually talk to.
Okay, so the best way to think about the ECS is like it's the master volume control for your brain. Its number one job is to maintain balance, what scientists call homeostasis. It's constantly working to make sure brain activity isn't firing too frantically or too slowly. It's trying to keep everything humming along in that perfect Goldilocks zone.
Now, this system has a few key parts. We've got CB2 receptors on immune cells, and our body even makes its own cannabinoids on demand. But for our story, the real star of the show are the CB1 receptors. And the truly wild part, the thing that makes them so special, is where they're located. You see, unlike most receptors, they sit on the terminal of the neuron that's sending the signal, and that weird placement is the secret to one of the brain's coolest tricks.
That trick is called retrograde signaling. Picture this. It's like the receiving neuron is sending a text message backwards to the neuron that just sent it a signal. So if neuron B is getting totally overwhelmed with signals from neuron A, it just whips up some of its own cannabinoids, sends them back across the gap, and they tell neuron A, hey, that's enough, calm down a bit. It's this beautiful on-demand braking system that keeps our brain's communication incredibly precise. All right, so our brain has this beautifully balanced system. What happens when we introduce outsiders? Well, let's meet the two main players from the cannabis plant, THC and CBD. They both knock on the door of the ECS, but believe me, they do it in profoundly different ways. First, we've got THC. This is the compound famous for the high. And you can think of it like an imposter key. It's a fantastic mimic, right? It slides right into the CB1 receptor's lock, a perfect fit, just like our body's own cannabinoids. But here's the catch. It completely ignores that precise on-demand system. So instead of a targeted little calm down text message, THC just blasts the signal everywhere, all at once, turning down the volume on neurons all across the brain.
And this really gets to the nitty-gritty of how they're so different. THC is what's called a partial agonist. That just means it directly turns the key in the CB1 lock. But then you look at CBD. It's called a negative allosteric modulator. It's a totally different strategy. It doesn't even try to turn the key. It does something much more subtle to the lock itself. And you'll notice it also talks to completely different systems, like our serotonin system, which is huge for things like anxiety.
So if THC is that imposter key trying to jam the lock, CBD is something else entirely. It's more like a master locksmith. It doesn't try to force its way into the keyhole. Instead, it attaches to a different spot on the receptor and actually changes the shape of the lock. And by doing that, it makes it much harder for THC's key to get in and do its thing. So in a way, CBD can act like a chaperone or a buffer, toning down THC's overwhelming effects and helping restore a little bit of that balance.
And that brings us to the other side of this paradox. What happens when that delicate, precise volume control system in your brain is constantly being hijacked and overwhelmed by that THC imposter key?
And here's a number that is absolutely crucial to understand. The cannabis people are using today it's not your parents' cannabis. Not even close. Back in the day, the average THC potency hovered around 3%. Today, the average is over 12%, and you can easily find products that are way, way stronger. That means the brain is being hit with a signal that is orders of magnitude more powerful than what it evolved to handle.
Now, this is especially dangerous for the adolescent brain, which is basically a massive construction site. The ECS is one of the key architects during this phase. Chronic, heavy THC use can actually speed up the thinning of the prefrontal cortex. Think of that as the CEO of your brain, right?
It's in charge of planning, impulse control, the whole shebang. The research shows that this overload can also reduce how well important brain networks talk to each other. And we're not just talking about temporary brain fog. The evidence points to long-lasting, durable changes.
Okay, so what about that really scary link to psychosis? How does that even work? Well, it's a super fascinating kind of backwards process. Remember, THC's main job is to inhibit things, to turn the volume down. But in certain brain circuits, it ends up inhibiting the inhibitor. It silences these little GABA neurons, whose entire job is to act as the brakes on your dopamine system. So THC basically takes the foot off the brake pedal, letting dopamine flood the system. And that dysregulation of dopamine is a hallmark feature of psychosis.
Now, faced with this constant onslaught of THC, your brain does something pretty smart. It starts removing its own CB1 receptors to protect itself. It's called downregulation, and it's the reason people build up a tolerance. But, and this is a really hopeful but, it doesn't have to be permanent. The brain is incredibly resilient. Studies show that after about 4 weeks without THC, the brain can rebuild its supply of CB1 receptors right back to normal levels. The system can absolutely heal itself.
Okay, so we've talked a lot about the risks of just flooding the system. But what if instead of a flood, we used a gentle, precise touch? This is where we flip the coin and look at the reward side of the paradox, cannabis as a source of incredibly powerful medicine.
Because when you isolate these compounds and use them with surgical precision, they can be absolute game changers. We see it with purified CBD, which is an FDA-approved drug that calms the storm of seizures in kids with rare forms of epilepsy. We see it in balanced THC/CBD medicines that help MS patients with debilitating muscle spasms. We even see the promise of CBD for anxiety, where it's not even hitting the ECS, but interacting with serotonin receptors. You see the pattern? In every single one of these success stories, the key word is precision.
All right, so here we are. We've seen the risks of using a blunt instrument, and we've seen the incredible rewards of a surgical tool. So how do we finally make sense of it all? How do we resolve this paradox?
And it all comes down to this one simple idea. The plant isn't the hero, and it isn't the villain. It's neutral. The real difference between something being a medicine or a risk, it's all in the application. It's about which specific compound you're using, the dose, the ratio to other compounds, and even the timing. That's where the magic and the danger really lies.
So think of it this way one last time. High THC recreational use, that's taking a sledgehammer to a complex, delicate system, causing widespread, unpredictable effects. But targeted cannabinoid medicine, that's like a neurosurgeon's scalpel, aiming for a very specific pathway to get a very specific and desirable outcome.
And that really leaves us with the big question, doesn't it? As science gets better and better at pulling apart these compounds, the old debate of, is cannabis good or bad, well, it's becoming obsolete. The real question, the one we all have to grapple with now, is this. How do we harness the incredible precision of this plant's chemistry while being smart enough to avoid its very real pitfalls?
Welcome to the Deep Dive. Today, we're really getting into the weeds, not just on cannabis itself, but on the fundamental brain system it interacts with. That's the endocannabinoid system, or ECS. Think of it as the brain's master regulator. And, understanding this system is really the only way, I think, to truly get the difference between, say, THC and CBD at a neurochemical level.
Absolutely. It's crucial.
Our mission today, we want to shift the conversation away from just the plant, the strains and products, and really dive into the pharmacology, what's actually happening in the brain. We're drawing on a whole range of research here in neurobiology, how these compounds move through the body, developmental impacts. It's a pretty comprehensive look.
And it's fascinating stuff.
Yeah.
Because to understand why cannabis affects you the way it does, you first need to appreciate this incredibly sophisticated internal balancing act your brain performs constantly.
Like a safety net.
Exactly, a safety net. And THC doesn't just, tap into it. It kind of overwhelms it.
Okay, let's start there then. The safety net, the ECS. What exactly is it? You said it's about balance, homeostasis.
Right. It's an endogenous system, meaning your body makes its own components. It's primarily made of lipids, fats, and its main job is regulating synaptic activity, keeping things stable.
And you mentioned the CB1 receptor earlier. That's a big part of this.
Huge. It's actually the most abundant G protein-coupled receptor, or GPCR, in our entire central nervous system. That's massive.
Wow. So it's everywhere in the brain.
Pretty much everywhere, yeah. And that's key to why THC's effects are so widespread, so psychoactive. There's also CB2, but that's mainly found peripherally in immune cells involved more in inflammation.
Okay, so CD1 is the main brain target, and the body makes its own keys for these locks, right? The endocannabinoids.
That's right. The two main ones we talk about are anandamide, or AEA.
Also called the bliss molecule.
Sometimes, yeah. And the other is 2-arachidonyl-glycerol, or 2-AG. And here's something really interesting from the research. The baseline level of 2-AG in the brain is something like 1,000 times higher than AEA.
1,000 times. Okay, so 2-AG is really running the day-to-day operations then.
It seems to be the primary workhorse, yes. It's a full agonist at CD1, meaning it activates it strongly, and it's deeply involved in synaptic plasticity, how connections strengthen or weaken.
And anandamide, AEA.
It's only a partial agonist at CV1, so weaker activation there. But interestingly, it's a full agonist at another receptor called TRPV1.
TRPV1, isn't that involved in pain and heat sensation?
Exactly. So AEA provides this intriguing link between the ECS and how we process pain or potentially noxious stimuli.
Okay, so we have receptors, we have the body's own keys. But you mentioned something about the system being unique and how it communicates, going backwards.
Yes, this is probably the most distinctive feature. It's called retrograde signaling. Normally, a signal goes from the sending neuron, the presynaptic one, across the synapse to the receiving postsynaptic neuron. Standard stuff. But with endocannabinoids, it's flipped. They are actually made on demand in the postsynaptic neuron, the receiving cell. Usually when that cell is getting a lot of input, getting highly activated.
So the receiver cell makes them when it's getting bombarded with signals.
Precisely. And then these lipid messengers, they don't wait around. They diffuse backward across the synapse.
Back to the sender cell.
Back to the presynaptic terminal, where they bind to those CB1 receptors we talked about.
Okay, so the receiving cell is literally telling the sending cell to, what, chill out.
That's a perfect way to put it. It's a negative feedback loop. When the endocannabinoid hits that presynaptic CB1 receptor, it triggers an inhibitory cascade inside the sending neuron.
Inhibitory, meaning it slows down neurotransmitter release.
Exactly. It depresses or reduces the release of whatever neurotransmitter that neuron was sending out. It's a very precise, localized break. It stops individual synapses from getting overexcited. Think DSI or DSE, depolarization-induced suppression of inhibition or excitation. It's this rapid response break.
A localized volume control? I like that.
But here's the critical point for today's discussion. Phytocannabinoids, like THC from cannabis, they don't wait for that internal too much activity signal.
They just jump in.
They just flood the system and activate CB1 receptors everywhere, whether they need breaking or not. It's a global, generalized inhibition that completely bypasses the brain's own finely tuned, localized control. It's like slamming on the brakes on every car on the highway simultaneously.
Okay, that makes sense. That distinction is huge. The brain uses it locally, THC uses it globally. So let's talk more about THC versus CBD now. How do they differ in interacting with this system?
Well, first let's touch on how they get around the body, the pharmacokinetics. As you mentioned, they're both very lipophilic, fat-loving.
Meaning they dissolve easily in fats.
Right, which lets them zip right across the blood-brain barrier. That's why the effects can come on quickly. But it also means they get stored in body fat.
And that affects how long they stick around.
Definitely. Especially for THC. If someone uses cannabis regularly, THC builds up in fat tissues. So even after they stop, it slowly leaches back out. The elimination half-life can be, 5 to 13 days in chronic users.
Days, wow. So the brain is potentially exposed for a long time after the last use.
Exactly. That persistent exposure is a major driver of the neuroadaptation we see, like tolerance. The brain is constantly dealing with this lingering inhibitory signal.
And how you take it matters too, right? Smoking versus edibles.
Oh, absolutely. Inhaling gets it into the bloodstream and brain very fast peak effects in maybe 3 to 10 minutes. Bioavailability is pretty decent too, maybe 10–35%.
But edibles are different.
Much different. When you eat it, it has to go through your digestive system. Then the liver, that's called first-pass metabolism. The liver breaks down a lot of the THC before it even reaches the bloodstream.
So less gets through and it's slower.
Much slower onset,
Yeah.
Maybe 1 to 2 hours to feel effects, sometimes longer. And the bioavailability is way lower, maybe only 4 to 12%. The experience is really quite different based on route.
Okay, pharmacokinetics covered. Now, the actual mechanism at the receptor level, THC, that's delta-9 tetrahydrocannabinol, that's the main psychoactive one.
Right. THC acts as a partial agonist at both CB1 and CB2 receptors. Partial means it activates them, but maybe not to the absolute maximum extent possible, like 2-AG might. But critically, it does activate them.
And because CB1 is everywhere, that activation causes that global depression of signaling we talked about, hitting the brakes everywhere.
Precisely. It reduces the release of both excitatory neurotransmitters, like glutamate, and inhibitory ones, like GABA. It's a widespread dampening effect.
Now, CBD, cannabidiol. This one's fascinating because it's non-psychoactive. It doesn't cause a high. How does it work?
It's completely different from THC. CBD has very low affinity for the main binding site on the CD1 receptor where THC docks.
So it doesn't directly activate it like THC does?
No. Instead, the prevailing theory, and there's good evidence for it, is that CBD acts as a non-competitive negative allosteric modulator, or NAM.
Okay, NAM, that sounds complicated. Non-competitive negative allosteric modulator. Break that down.
Let's try. Non-competitive means it binds to a different site on the CB1 receptor, not the main one THC uses. It's an allosteric site, like a secondary docking port.
Okay, a different spot.
Negative modulator means that when CBD binds to this other spot, it subtly changes the shape or conformation of the entire receptor protein.
It, like, twists it slightly.
Sort of, yeah.
Oh.
And that change makes it harder for the main agonists like THC or the brain's own AEA to bind effectively to their primary site. Or if they do bind, they don't activate the receptor as strongly.
Ah. So CBD doesn't block the main door, but it makes the door harder to open or less effective when it is opened.
That's a great analogy. It reduces the efficacy and the potency of agonists like THC. It essentially acts like a dimmer switch or maybe a safety catch, dampening the signaling through CB1.
Which explains why CBD can counteract some of THC's effects. It's literally acting as a buffer at the receptor level.
Exactly, a neurochemical buffer. And that's just its action at CB1. CBD is known to interact with a whole host of other targets in the brain, too.
Like what?
Well, it has significant effects on serotonin receptors, specifically the 5-HT1A type. Agonism there is thought to contribute to its anxiolytic, anti-anxiety, and possibly antidepressant effects.
Interesting.
And remember TRPV1 involved in pain. CBD also interacts with those, similar to AEA, which likely plays a role in its pain-regulating properties. So its therapeutic potential is much broader than just modulating CB1.
Okay, this distinction is really clear now. THC global break via direct partial agonism. CBD subtle buffer via negative allosteric modulation, plus other targets. Let's talk acute effects. If THC slams the global break, what does that do immediately? You mentioned memory.
Right, the hippocampus is a key area. It's dense with CB1 receptors. And it's critical for forming new memories, particularly through a process called long-term potentiation, or LTP.
LTP, that's the strengthening of connections between neurons, right? How memories get consolidated.
Exactly. It requires precise timing of signaling. THC's generalized inhibition really messes with that delicate process. By reducing neurotransmitter release, it can impair the strengthening of those synaptic connections.
So impaired memory consolidation. But the sources mention something surprising, a biphasic effect. Low doses might enhance LTP. How does that work if THC is inhibitory?
Ah, yes, the complexities of brain circuits. It likely comes down to disinhibition.
Disinhibition, inhibiting an inhibitor.
Precisely. Neurons don't exist in isolation. You have excitatory neurons using glutamate and inhibitory interneurons using GABA. CB1 receptors are found on both.
Okay.
It turns out that at lower concentrations, THC might preferentially bind to and inhibit the GABAergic interneurons slightly more effectively.
So it quiets down the neurons whose job it is to quiet down others.
You got it. If you silence the silencer, the net effect is a temporary increase in the activity of the main excitatory neurons. You remove the local GABA brake, allowing for potentially stronger signaling and maybe enhanced LTP, but only within a narrow low-dose window.
But only temporarily and only at low doses.
Right, because as the concentration of THC increases, that preferential effect is lost. The inhibition becomes global. It starts hitting the main excitatory glutamate synapses directly, overwhelming any disinhibition effect. And that's when you get significant LTP disruption and clear cognitive deficits like memory impairment.
And this links directly to modern cannabis, doesn't it? The THC concentrations today are often much higher than they used to be.
Dramatically higher. You know, back in the 60s, 70s, even 80s, typical cannabis might have had, what, 2%, 3%, maybe 4% THC?
Very low.
Now, it's routine to see strains with 15%, 20%, even 25% THC or higher, especially in concentrates, and often with very little CBD.
So users are easily pushing past that narrow potential disinhibition window and straight into the zone of significant synaptic inhibition and cognitive impairment.
Almost certainly yes. particularly with high-potency products. Which brings us back to CBD as the buffer. The research is pretty consistent here. Co-administering CBD alongside THC tends to blunt some of those acute negative effects.
Less memory impairment, potentially less anxiety or paranoia.
Generally, yes, because CBD, through that NAM mechanism, is tempering THC's ability to fully slam that global brake.
Yeah.
It really underscores that the THC and CBD ratio is probably the single most important variable determining the acute neurocognitive risk profile of a cannabis product.
Okay, so the ratio is key for acute effects. What about long-term chronic use? What happens when the brain is constantly dealing with that THC-induced global inhibition day after day?
The brain adapts. It tries to regain balance. The primary way it does this in response to chronic high-dose THC is through CB1 receptor downregulation.
Downregulation, meaning fewer receptors.
Fewer receptors, and the ones that remain might become less sensitive to activation. The brain is essentially turning down the volume on the ECS pathway because it's being artificially overstimulated by THC.
This drives tolerance, right? Needing more THC to get the same effect.
Exactly. That's the physiological basis of tolerance. It can also contribute to dependence and withdrawal symptoms when use stops, as the brain's natural ECS signaling is now dampened.
But you mentioned earlier there's some good news regarding reversibility. This downregulation isn't necessarily permanent.
That's a really crucial finding from human imaging studies, PET scans specifically. They've shown that in most brain regions, particularly the cortex, this CB1 receptor downregulation is reversible.
How long does it take?
The data suggests that after about 4 weeks of continuous abstinence, CB1 receptor density typically returns to levels comparable to non-users.
4 weeks? That's actually quite hopeful. It means the brain can reset that aspect of the system if the drug exposure stops.
It is hopeful, yes. It shows the dynamic nature of the system. But, and this is a big, but this reversibility might not tell the whole story, especially when use starts during adolescence.
Ah, yes. Adolescence is always highlighted as a period of heightened vulnerability. Why is that in the context of the ECS?
Because during adolescence, the brain isn't just maintaining itself, it's undergoing massive construction and refinement. And the ECS plays a critical role in guiding some of those processes.
Like what?
Things like synaptic pruning, the process of eliminating unnecessary or weak connections to make brain circuits more efficient. Also myelination, development of executive functions in the prefrontal cortex, impulse control. The ECS is involved in modulating all of this.
So introducing a powerful ECS disruptor like high-dose THC during this critical construction phase, that's problematic.
Highly problematic. It's like constantly interfering with the architect and the construction crew while they're trying to build a complex structure according to a specific blueprint.
What does the research show in terms of actual impact? Are there measurable differences in the brains of adolescents who use heavily?
There are, yes. Some longitudinal studies following individuals over time have linked heavy adolescent cannabis use with structural changes, most notably accelerated cortical thinning, particularly in the prefrontal cortex, PFC.
The PFC, that's the hub for planning, decision-making, judgment.
Exactly. The thinning suggests that the normal ECS-guided synaptic pruning process might be disrupted, potentially leading to less efficient or structurally altered circuits in this really critical brain region.
Less efficient. Does that show up in how their brains function, too?
It often does. Functional MRI studies, looking at brain activity during cognitive tasks, sometimes find that adolescent users show increased brain activation in task-relevant areas compared to non-users, even if they perform the task equally well.
More activation. Wouldn't that mean their brain is working better?
Not necessarily. It's often interpreted as a compensatory mechanism. Their brains might have to work harder, recruit more neural resources to achieve the same level of performance. It suggests reduced neural efficiency, like needing to rev the engine harder to get the same speed.
That makes sense. Less efficient hardware requires more effort.
And there's another layer, too. Some research suggests that vulnerability might sometimes precede the drug use.
What do you mean?
One study found that lower baseline functional connectivity, weaker communication in certain brain networks before heavy use began, specifically involving areas like the anterior cingulate cortex and orbital frontal cortex, actually predicted who would later go on to use more cannabis.
So pre-existing differences in brain wiring might make someone more susceptible to starting or escalating use.
It's a possibility, yes. It suggests a complex interplay where underlying neurobiology might increase risk, and then the drug use itself further impacts brain development. It's not always a simple cause and effect story.
Okay, that covers chronic use and the specific risks during adolescence. Now let's tackle something that often seems paradoxical, the link between cannabis use and psychosis.
Right. The epidemiological evidence is pretty clear. Heavy cannabis use, especially high-potency THC used frequently from a young age, is associated with an increased risk of developing psychotic disorders like schizophrenia. The odds ratio is often cited around 2, meaning roughly double the risk, particularly in genetically predisposed individuals.
And there seems to be a dose-response relationship, more use, higher risk.
Correct. But this leads to that apparent contradiction we touched on earlier.
Yeah, if THC is fundamentally inhibitory, slamming the brakes, how does it increase the risk of psychosis, which we usually associate with overactivity, particularly in the dopamine system?
That's the crux of the issue. The leading hypothesis to explain this involves dopamine dysregulation. Despite its overall inhibitory nature, THC does seem to enhance dopamine signaling in key brain regions like the striatum.
It increases dopamine release or activity, but how?
Again, it comes back to that concept of disinhibition and the specific circuits involved. Remember, CB1 receptors are densely located on those inhibitory GABA interneurons.
Okay.
In dopamine pathways, these GABA interneurons normally act as gatekeepers, putting a brake on the dopamine-releasing neurons.
They keep dopamine in check.
Exactly. Now, introduce THC. It binds to the CB1 receptors on those GABA interneurons and inhibits them.
So THC inhibits the inhibitor of dopamine.
Precisely. You silence the silencer. By removing the GABAergic brake, THC effectively disinhibits the dopamine neurons, allowing them to fire more and release more dopamine into the synapse.
Wow. So the net effect in that specific pathway is increased dopamine activity, even though THC's overall action across the brain is inhibitory.
That's the thinking, yes. It resolves the paradox. THC slams the brakes globally, but by hitting the brakes on the GABAergic gatekeepers in the dopamine system, it paradoxically causes the dopamine accelerator to be pushed harder. And excessive dopamine activity is strongly implicated in the positive symptoms of psychosis.
That makes so much more sense now. It's about which brakes it hits hardest in specific circuits. Okay, we've covered the risks and the mechanisms quite deeply. Let's pivot to the therapeutic side, because cannabis compounds are used medically.
They are, and there's real potential, but the key here is precision. Unlike recreational use, which is often like using a pharmacological sledgehammer, therapeutic success hinges on isolating specific compounds and using precise, controlled dosing.
Can you give some examples?
Sure. The clearest successes have come from CBD. There are FDA-approved, purified CBD medications, like Epidiolex, for treating severe childhood epilepsy syndromes, like Dravet and Lennox-Gastaut.
And is that working via the CB1-NMN mechanism?
Probably not primarily. The thinking is that CBD's anti-seizure effects in these cases are likely mediated through its other actions, perhaps reducing neuroinflammation, modulating calcium channels, or interacting with other receptors, not mainly by buffering CB1.
Interesting. So its other targets are key there. What about THC in medicine?
THC also has therapeutic uses, often in combination with CBD. Sativex, for example, is an oral spray with a roughly 1.1 ratio of THC to CBD.
What's that approved for?
It's approved in many countries for treating muscle spasticity, like stiffness and spasms associated with multiple sclerosis, MS, particularly when other treatments haven't worked.
And there, the THC's CB1 action is probably important for muscle relaxation.
Yes, in that case, the direct CB1 agonism from THC is likely contributing significantly to the therapeutic effect on spasticity, while the CBD might help mitigate some side effects and potentially add its own anti-inflammatory benefits.
So it really depends on matching the compound's specific mechanism to the specific disease pathology.
Absolutely. You need the right tool for the job. And this highlights the major challenge facing the broader field of cannabinoid therapeutics right now.
What's the main barrier?
It's largely the lack of standardization and rigor in clinical trials. For many potential applications, we're still looking at mostly smaller Phase 2 studies. There's huge variability in the products. Use different ratios, different extraction methods, inconsistent dosing, and different ways of measuring outcomes.
So it's hard to compare results or draw firm conclusions.
Very hard. To get widespread clinical acceptance and regulatory approval, we need large-scale, well-controlled Phase 3 trials using standardized, characterized products with clear dosing regimens. Until then, a lot of the potential remains just that potential. Clinical success absolutely demands that precision.
Right. Okay, let's try to summarize the key takeaways from this deep dive. We've seen the ECS as this crucial internal regulator using retrograde signaling as a localized brake. THC essentially hijacks the system, acting as a global brake override through partial CB1 agonism. CBD acts differently. As a subtle buffer, a negative allosteric modulator, dampening CB1 signaling and hitting other targets too.
Correct.
Acutely, THC's effects, especially on memory, follow a biphasic curve due to disinhibition at low doses, but clear inhibition at higher doses, which are common today. The THC-CBD ratio is critical here.
Absolutely key for acute risk.
Chronically, THC leads to reversible CB1 downregulation, driving tolerance. But use during adolescent brain development poses unique risks, potentially disrupting structural formation and reducing neural efficiency.
A critical window of vulnerability, yes.
And the psychosis link, seemingly paradoxical, is explained by dopamine dysregulation via disinhibition, THC, silencing the GAGergic brakes on dopamine neurons.
A complex but elegant explanation.
Therapeutically, precision is everything isolating compounds and matching mechanisms to the pathology, as seen with CBD for epilepsy and THC-CBD for MS spasticity.
That's the path forward for medicine.
So, reflecting on all of this, particularly the distinct roles of THC and CBD, what's the final provocative thought you want to leave our listeners with?
Well, we've established CBD's role as that safety clip, that negative modulator at CB1, capable of buffering THC's effects. We also know THC potency is rising, increasing risks. So the big question I think we need to grapple with is this. Given what we know, could, or maybe even should, precisely dosed CBD be strategically employed, perhaps even mandated in regulated markets, as a specific neurochemical protective agent? Could we intentionally leverage its buffering capacity to mitigate the known acute cognitive and potentially psychotic risks associated with the high-THC products readily available today?
Using CBD not just therapeutically for specific conditions, but prophylactically as a harm reduction tool alongside THC.
Exactly. Is that a viable path forward for public health? It's complex, ethically and practically, but given the pharmacology we've discussed, I think understanding CBD's potential as a targeted risk mitigator is where a lot of future research and policy debate really needs to focus.